when we come to bioremediation, I think the world now needs a lot of focused area of work, research, interdisciplinary and technology intervention for this type of work, which is completely industrial oriented. Now I invite Professor Mautisi Mitra Madam to deliver her talk and enlighten us in both the topics. Thank you, Madam.
Thank you very much. First, let me go and share my screen. Okay, now I'm going to activate my laser pointer. Okay, everybody can see my pointer, right? Okay, now I want to minimize. Just a minute. Okay, good morning to all of you. And I first would like to thank all of you at the Centurion University of Technology and Management for inviting me to present my research on this 36th National Science Day. Because I looked it up that India started celebrating the National Science Day since 1986. And I am excited and feel very honored. You guys have already showered a lot of praise on me and I'm feeling overwhelmed. But I would like to apologize to all the students, and also the faculties who had to get up so early morning Indian time zone to attend this webinar. I am really impressed with you guys. So, as many of you have said, that this year's theme is Integrated Approach in Science and Technology for Sustainable Future, right? So as you can read from the title of my research, it has some flavor of it for, that reflects the theme of the 2020 National Science Day. So first, I'm going to give you a short intro of myself. I'm from Kolkata, from West Bengal. I did my Bachelor's of Science in Botany from Presidency College, then got my master's degree from Balangir Science College, Botany Department. And then in 1996, 26th December, I moved to LSU, Louisiana State University in Baton Rouge, Louisiana, to do my PhD in plant biology. And after I received my PhD in plant biology, I moved to UC Berkeley in California to conduct my postdoctoral research. So both at LSU and UC Berkeley, I'm a plant biologist. So my research topic was light and carbon dioxide acclimation of photosynthesis in the model microgreen alga Chlamydomonas reinhardtii. I never worked with bacteria before formally, to be honest, except cloning genes in E. coli, Chlamydomonas genes in E. coli, and teaching microbiology labs to the undergrads. That's all I had experience about microbiology and also some coursework, right? And then in 2009, I got this tenure track assistant professor position at University of West Georgia in Carrollton, which is a rural area in Georgia, 45 miles from Atlanta, which is a big city. Now, University of West Georgia is primarily an undergraduate institution with master's program. We don't have PhD program. So let me tell you from the very beginning, my research colleagues, my team is comprised of undergraduate research students, high school research students, and master's students. So having given that brief intro, I am going to go to the talk. So this will be my lecture outline today. First, I'm going to introduce you to Clammy and how we isolated the two bacterial strains, followed by published results on these two bacterial strains. Then I will talk about some ongoing experimental results, which are unpublished, and end with a future experimental plan. It's a kind of long talk, I hope I don't bore you to death. And I expect you to ask me any kind of questions after my talk is over, or if you want to ask in the middle, that's fine with me too. So, as I said, I'm a plant biologist and I work with Chlamydomonas my whole life. And I called Chlamydomonas fondly as Clammy baby, by the way. So in my talk, I'll refer Chlamydomonas as Clammy, okay? Now, this is how my pointer is pointing to the Chlamydomonas cell appears under the microscope. So it has a large cup-shaped chloroplast, which fills 80% of the cell volume. You see this orange spot? This is the eye, or the eye spot of Chlamydomonas loaded with carotenoids. So it can sense just light, just like our eyes can. It has two flagella to swim around. So pretty much the phototaxis of this alga is mediated by light reception by the eye spot, and then an action potential or electrical current is generated, which is passed to the base of the flagella, and the flagella beating pattern changes according to the electrical impulse, which allows the cell either to move towards the light or away from the light. And you must have heard that, already know about this field probably, the modern biomedical field, you can say, the optogenetics, which actually opened up a few years back when the gene, channel rhodopsin gene from the eye spot of this alga, which is a blue light activated sodium channel, was cloned into the hypothalamus of the mouse, to make the neurons light sensitive, so that neuroscientists can manipulate neuron clusters in the different areas of the brain to do functional mapping. So that research pretty much came because of this channel rhodopsin gene from the eye spot of the clammy. And it has been also used for treating blindness in human. We have an FDA approval on that channel rhodopsin gene. And the genes that control the flagella development of clammy, humans, we have those genes. Why? Because we have cilia, the sperm cilia, lung alveoli cilia, brain ventricule cilia. So the genes responsible for the cilia development these are identical to the genes that are responsible for the flagella development in clammy, because the structure of flagella and cilia are the same. So there are many medical conditions that can happen because of defect in this flagella genes. And so scientists, biomedical researchers use clammy as an experimental model system to test drugs, to pretty much study these genes and how they play a role in cilia development to come up with cure for many genetic conditions in humans. So clammy is not only a model system for plant molecular biologists like me, but it is also a model system for medical scientists and bioenergy researchers. So in the lab setting, we grow clammy on a media called Dept, which contains tris, acetate, phosphate. Acetate is the carbon source. So clammy is very unique compared to higher green plants in the sense it can behave as an autotroph or it can behave as a heterotroph depending on whether it gets acetate in the nutrient media or not. So if there is acetate present in the growth media, it will go for the acetate, use it up to convert it into sugar via the glyoxylate cycle, which plants and many bacteria have. And if the acetate is not there in the growth media, it has to make glucose via photosynthesis. So often we call clammy as a green yeast, that's why. Now, today's topic is about bacteria. So how come a plant biologist like me ended up with a bacterial project that you saw in the title, right? So there's a short story behind it. Since we grow clammy on acetate containing media, sometimes we do get bacterial contamination on the Dept media plate. Like you see in these two pictures, these are two different clammy strain culture plates, Dept plates, and you see some yellow bacterial contamination on both plates, right? So usually when we get contamination, so you can say, why did you have contamination? Because bacteria can also use, many bacteria, they can use acetate as a carbon source, just like Clammy. So we usually throw these plates away in the trash can, pretty much. But in fall 2018, a high school student from the local high school walked into my lab and wanted to do research. And she's a high school kid. She will come after school hours, after 2 p.m., and will stay for like 2 to 3 hours, maybe maximum. So I thought, what kind of project I can give her? So at that time, we were having this bacterial contamination, and I suddenly got this idea, okay, let's not throw these plates away. So I told that student, I can give you a simple project. You have to purify the 2 bacterial strain from these 2 contaminated plates. And I purify the bacteria, and then I will give you 5 antibiotics that are in my lab, and you can test 2 different amounts of a dose of those 5 antibiotics to see which one will kill the bacteria, but not my clammy cells. So the whole project started as a mere, maybe short span high school student science project. I never in my wildest dream, I would have thought that this project will become my main project and the Clammy project will be at the back burner. So the high school student project started like this with the 2 goals: purify the bacterial strains and identify antibiotics that will kill the bacteria, but not Clammy. So she purified the bacteria using streak plate method, and you can see the 2 yellow pigmented bacteria, which we named as CC4533 and CLIP185, based on the Clammy strain name, a strain plate they contaminated. So these are strain names of Clammy actually, because we didn't know the identity of the bacteria, at that time, because this is how the project is evolving. So, and these plates are of LB plate. LB is a very rich nutrient to grow bacteria. I think many of you know that. So this is a table that shows you a bunch of numbers with standard deviation. And it's actually the diameters of zones of growth inhibition in the disc diffusion antibiotic susceptibility test. So what that high school student did was, she tested 2 different amounts, 50 microgram and 100 microgram of these 5 antibiotics, and measured the diameters of the zones of growth inhibition. So of course, she did replicates. You have to do replicates in a science experiment. So the logic is, the larger the diameter of zone of growth inhibition on that antibiotic plate is, the more susceptible the bacteria is to that antibiotic dose. So as you can see, I have highlighted in red a neomycin amount, 50 and 100 micrograms, which produce the largest diameter for CC4533. That means CC4533 is very susceptible to neomycin, although Clammy also gets affected, but not as much as CC4533. But for CLIP185, vancomycin turned out to be the best antibiotic, actually. If you see the numbers here, it has the largest diameters for the zones of growth inhibition. By the way, neomycin affects protein synthesis, a translation step in bacteria, and vancomycin affects the cell membrane of the bacteria. So based on the disc diffusion antibiotic susceptibility test, we decided that neomycin and vancomycin are the best antibiotics to eradicate bacterial contamination from Clammy plates. But as a scientist, we do need to check our results. So what we did, we wanted to confirm the efficacy of neomycin and vancomycin. So we streaked both Clammy and the individual bacteria on tap media plate containing 50 micrograms per mil of neomycin and 50 micrograms mils of vancomycin. And we allowed the cells to grow for a week, just to make sure that there will be no bacterial colonies showing up after the incubation period. And as you can see, there are no traces of bacterial growth on any one of these 2 plates. So we found, that was the project of that high school student, and she has found the 2 drugs and the dose to be used to eradicate contamination on my clammy culture plates. But the high school student still got so interested, and she wanted to do some biochemical test along with my undergraduate students. So we started characterizing biochemically these two bacterial strains. So obviously, any microbiology student knows the first thing you have to do is to do a gram stain. So we did the gram stain, and CC4533 is a rod, gram-negative rod, large rod, and CLIP185 turned out to be gram-positive, small rod. Then we also checked for the temperature preference for growth for both CC4533 and CLIP185 on the LB medium and the TAP medium. And we tested 3 different temperature. Just let me go do one thing because it's irritating me. I'm going to hide the floating controls. Okay. So as you can see from my the slide, CC4533 grows well on LB as well as the algae medium. But there is a pigment deficiency when it's growing on TAP. Look at the color, okay? But it is very sensitive to higher temperature. It grows best between 22 to 30 maybe. So around 37, it cannot grow. And another thing we noted with increasing temperature, there is also pigment deficiency that's happening even on the LB media. So just to be sure that the temperature is a problem for pigment biosynthesis in CC4533, we spotted the liquid culture of CC4533 on TAP and LB plate and incubated at 35C, not 37C. And we saw it's completely bleached. I mean, there's no pigment at all. It's white. It's growing, but it's white. We shifted that same plate back to room temperature, and you can see the pigment accumulation happening at room temperature. This experiment clearly showed that high temperature is not conducive for pigment accumulation in CC4533. For CLIP185, we found out that it grows very nicely on the bacterial media, but at any temperature between 22 and 37 C. But it was a surprise when we looked at the growth on the TAP plate. It's struggling to grow on the TAP media, which is the algae media. And it's surprising. Why? Because we isolated it from a contaminated clammy culture plate, which was a TAP media plate. But by itself, when we streak it without clammy on the plate, it is struggling to grow. We found that very interesting, but we didn't understand why it was doing that. We thought maybe it was getting something from clammy. Then only it can grow on the TAP media plate, but that was beyond the scope of our research project. We didn't pursue. But I will show you one piece of experimental data at the very end. We now know why it cannot grow on TAP. That answer, we have it now. So anyway, let's move ahead. So next we checked actually if these two strains can use alternative carbon source because like glucose, sucrose, and lactose. So what we did is we formulated this medium called TP medium. TP simply means TAP medium without the carbon source acetate. So no bacteria will grow on the TP medium because there's no carbon source on it. So A, C, and E, these pictures, these are TP medium plate where we streaked CC4533 on it. And this medium also we added a phenol red, a pH indicator, which turns yellow in acidic condition and bright pink in alkaline condition. So we wanted to see if CC4533, when it is streaked on TP supplemented sugar plates with the phenol red, is it able to ferment these sugars into acid? Because the acid will then change color of the phenol red to yellow. And as you can see, yes, CC4533 is able to ferment glucose, sucrose, lactose into acid, which changes the phenol red, reddish color to yellow. Then we also streaked CC4533, since it's a gram-negative bacteria, on mcconkey agar, which is commonly called MAC. Now, MAC contains lactose, bile salts, crystal violet, and neutral red, which is a pH indicator. So neutral red in acidic pH turns pink, and in alkaline or pH, it turns kind of colorless or yellowish, actually. So this mcconkey agar test is used in microbiology as a selective, to select for gram-negative bacteria, and also to differentiate between gram-negative bacteria that can ferment lactose and that which cannot ferment lactose. So since CC4533 is a gram-negative bacteria, we thought we'll test it out. But just to help you to interpret the results, if you have not done a mcconkey agar test, that is, if the bacteria ferments lactose, it will produce acid and the color of the neutral red will change to pinkish. And if it doesn't, the plate becomes colorless from its reddish color or yellow, like light yellow. So when we did the mcconkey agar test, we found that CC4533 is not growing on it at all. But we earlier showed that it can ferment lactose, because from the TP plus lactose media experiment that I showed you, it ferments lactose. So why is it not growing? Because this media has bile salts and crystal violet, which actually only the enteric bacteria like E. coli that thrives in our gastrointestinal tract can tolerate. Non-enteric bacteria won't grow on it, although they might be gram-negative. So CC4533 probably is inhibited because of the bile salt and the crystal violet, although it can use lactose, we know, because from a different experiment. So we determined that it is a non-enteric bacteria. We did the similar sugar, TP supplemented with sugar tests for Clyp185. But as you can see from the picture B, D, and F, which are the sugar supplemented TP plates with the phenol red, and there's hardly any growth. And it's not surprising because it cannot grow on tap, as I showed you very well. Although it's still growing, but not much, and it is fermenting the sugar too. But there was no growth on lactose. So next test we did was to check if these bacteria are producing hemolysin, which are enzymes that rupture red blood cells or degrade hemoglobin. So in order to do this test, you have to streak the bacteria on tryptic soy blood agar, which contains 5% sheep blood in it. And based on the nature of the growth, like you see 3 signs, Greek letter sign here, alpha, beta, and the gamma. So based on the growth pattern on tryptic soy agar, we can classify the type of hemolysis. If the hemoglobin is incompletely degraded, producing a dark halo or a greenish halo around the growth of the bacteria, this outline, then it's alpha hemolysis. If it completely degrades the hemoglobin and produces a transparent clear zone around the growth of the bacteria, then it's beta hemolysis. And that's what you have when you have strep throat, tonsil infection, those are all streptococcus, those are beta hemolytic strains. And then gamma hemolysis means no hemolysin secretion, so no red blood cell destruction, and the bacteria just grows, but there is no such discoloration of the media or complete, clear zone around the growth. So we streaked CC4533 and Clyp185 on the tryptic soy blood agar. And as you can see, I hope you can see the dark halo around the growth. So both strains are alpha hemolytic. As a control, we had a beta hemolytic Staphylococcus aureus strain there. And you can see the clearing of the zone there around the growth. Now, we did a test for the presence or absence of cytochrome C oxidase. Now, if you've taken biochemistry, and studied cellular respiration, cytochrome C oxidase takes electron from cytochrome C and gives it to oxygen in the electron transport chain of cellular respiration, right? So many bacteria uses cytochrome C oxidase, or sometimes they use other alternative oxidase in their electron transport chain. So if a bacteria has cytochrome C, of course, it does C oxidase, it is most probably aerobic or microaerophilic or facultatively aerobic. But if it lacks cytochrome C oxidase, it doesn't necessarily mean that they are anaerobic bacteria. So how do we test for this enzyme? We usually buy commercial strips, testing strips, like you see this white strip, which has reaction blocks in it. And these reaction blocks are coated with a redox sensor called TMPD. And if we streak the bacterial cell, each type, in one of these two blocks, and we incubate it for 2 minutes, not longer than 2 minutes, because time is critical for interpretation of the result of this test. And if cytochrome C oxidase is present in the bacterial cell, it will oxidize these redox sensor to a purple colored indophyllon blue compound within 2 minutes. And so you can see CC4533 is purple, whether CLIP185 stays as yellow. So it is oxidase negative, and CC4533 is oxidase positive. We also did the starch hydrolysis test. Many bacteria, they secrete the amylase enzyme, which can degrade external starch into glucose or dextrin, and they can take it up for energy production. So how do you do the starch hydrolysis test? You usually take the bacteria that you want to test on and streak it on a media that contains starch. We used Muller-Hinton, which contains 0.15% starch, and let the bacteria grow. So you make a streak and let it grow for 2 to 3 days. And then you take a picture of that plate. So we test streaked CLIP185, CC4533, and the Bacillus subtilis, which is known in the literature to be a starch hydrolysis positive strain. So we used it as a control. So A, C, and E are taken after the growth. And then we flood the plate with iodine. The starch test using iodine. So if starch is present, it will turn the iodine will turn starch into blue, black, or brownish in color, depending on the concentration of that iodine solution, really. So we flooded the plate A, C, and E with iodine and incubated it for 15 minutes, drained off the excess iodine, and we wanted to see if the entire media, if there is any clearing around the growth, like you see this arrow, there is a clearing around the growth on a CLIP185 plate and also Bacillus subtilis plate. This shows in that area, the vicinity around the growth, there is no starch. The rest of the plate has a light bluish or brownish tinge, but there is a clearing. That means there is absence of starch. That means the amylase has degraded the starch. So CLIP185 and Bacillus subtilis, they are starch hydrolysis positive. Whereas CC4533, you see universal like brownish, bluish halo, there's no clearing if you look very carefully. So CC4533 is starch hydrolysis negative. Since CLIP185 is a gram-positive bacteria, we wanted to streak it on mannitol salt agar to see if it grows or not, and if it's going to ferment mannitol present in the mannitol salt agar. Now, mannitol salt agar has not only mannitol, a sugar alcohol, as a carbon source, but it also has high amount of salt and the pH indicator phenol red. So this test actually is a selective and differential test. It selects for gram-positive bacteria and differentiates mannitol fermenters from mannitol non-fermenters. So you can see, as in this demo example, that staph aureus ferments mannitol, produces acid, so phenol red turns yellow, whereas staph epidermidis doesn't ferment mannitol, although it grows, and so the phenol red color is retained. So, hello? Okay, I thought somebody was asking me something. So we streaked CLIP185 and staph aureus on MSA plate, and we monitored the growth and the color change. As you can clearly see, that, let me move this up. Staph aureus within 3 days changed the color of the agar. So it's yellow. So it has fermented the mannitol very quickly. Compared to Clyp 185, which is fermenting mannitol, you can see the light pale yellow around the growth, but it's doing it very slowly, actually. Okay, I do not. So now coming to the molecular studies. So all of you know that 16S rRNA gene, the small subunit, ribosomal subunit gene, is a phylogenetic marker to study bacterial phylogenetic relationship. So the top figure A is a schematic diagram of the E. coli 16S rRNA gene. It has 9 conserved regions denoted by C1 through C9 and 9 hypervariable region where you expect frequent changes in the nucleotides. And then within the conserved region 4, C4 region, you have this black box in the figure, which represents a super conserved region where you usually don't find any nucleotide change at all. Now, let me explain one thing. The conserved regions are not really conserved region. You will find occasional nucleotide changes, but the nucleotide changes won't be happening that frequently like it does in the hypervariable region. So what we did is we designed 2 PCR primers, one on the C2 region and the other on the C4 region. And we use these 2 primers to partially amplify the 16S rRNA gene from CC4533 and Clyp 185. And then we sequenced the PCR product and we blasted it in the NCBI and we got a hit, which had a 99.5% sequence identity to Sphingobia unique strain PR86. Now, you must note it's not 100% identical because as you can see from the figure B, I have the schematic diagram of the CC4533 partial 16S rRNA gene sequence, and the nucleotides in red at those particular position number, which are the nucleotide position number 797, 798, and this zone, the super conserved region. We have like 2 nucleotide changes in the conserved region C4 in CC4533 with respect to the hit, which is Sphingobia unique strain PR86. But please understand, this is a very short region, about like 460 base pair, almost, in length. So at least we got a taxonomic hit. For Clyp 185, we got 2 hits, which had a high identity to 2 different Microbacterium strains. One is Microbacterium benoti, and the other is a Microbacterium species. And both had a sequence identity of 99%. For the Microbacterium benoti strain, if we compare its sequence 16S rRNA gene with the Clyp 185s, we see there are 4 nucleotide changes, 3 in the C4 region, and 1 actually is in that hyper-conserved region, and again, 1 in the C2 region. For the other Microbacterium species strain hit, we see 3 nucleotide changes. So now we have some handle on the project. We know the genus, Sphingobium and Microbacterium. By the way, Sphingobium belongs to the alpha proteobacteria group, and Microbacterium belongs to the actinobacteria group. So we started looking up in the literature to see, because we are street smart microbiologists. I'm not a trained like a bacteria person, right? And neither are my students. And so we actually did read a lot of papers, whatever information is available on Sphingobium and Microbacterium. And we found that some Sphingobium strains and Microbacterium strains, they make carotenoids. And all of you know, you just heard about your professor saying that carotenoids are, present in plants, algae, bacteria, and they are very strong antioxidants. They protect the cells from oxidative stress, and they help in the capture of solar energy, as in plants, helping in photosynthesis, and stabilizes membranes. And carotenoids have a big industrial market. They are used in photoprotective cosmetics and also as health supplements like beta carotene, lutein, zeaxanthin, and so on. So as these two bacteria were yellow pigmented, and of course they are Sphingobia and Microbacterium, but I'm going to still call them as CC4533 and CLIP185 till the end of my talk. So we wanted to know whether the CC4533 makes beta carotene or not. And we found in the literature there are Sphingobia that makes beta carotene and zeaxanthin. And for CLIP185, we found, because it belongs to the actinobacteria, some actinobacteria make this heavy C50 carotenoid called decaprinoxanthin. So we did extract the pigment from CC4533, and we used pure beta carotene as a control. And we did spectrophotometric analysis of the extracted pigment from CC4533 and the pure beta carotene. We used an absorption, a wavelength scan program from 400 to 600 nanometer. By the way, carotenoids absorb in the blue region of the spectrum, okay? Not in the red region, like chlorophyll absorbs both in the blue and the red region, but carotenoids only in the blue region. So you can nicely see how the three peaks of beta carotene overlays, it's aligned with the three peaks in CC4533. So definitely CC4533 produces beta carotene, which is a C40 carotenoids. Then we did the similar experiment for CLIP185. We extracted the pigment from CLIP185 and from another actinobacteria called Cocuria, which is known in the literature to make decaprinoxanthin. And as you can see from the absorption spectrum, those three peaks of Cocuria nicely aligns with the three peaks of CLIP185. Now, we also did a double confirmation of decaprinoxanthin production in CLIP185. How? We used another famous actinobacterial strain. I think you must have heard about this strain, Corynebacterium glutamicum. It's a patented bacterial strain used for amino acid production on an industrial scale. So it also makes a decaprinoxanthin, and you can see again in the absorption spectrum peaks of Corynebacterium glutamicum pigment extract nicely aligns with the three peaks of CLIP185. So definitely CLIP185, which is a Microbacterium genus, it's making decaprinoxanthin. Now, in the literature, we also found that Sphingobium, there are different strains of Sphingobium that can use nasty toxic chemicals, aromatic compounds, and saturated hydrocarbon as the sole carbon source. So we decided to test, if CC4533 being a Sphingobium strain, can it actually do that? So we use the TP media, which lacks any carbon source. So no bacteria will, CC4533 doesn't grow on TP media. But if you supplement TP media with a saturated chlorinated hydrocarbon like cyclohexyl chloride or a polyhydroxyalkanoate like PHB or polyhydroxybutyrate, which is a biodegradable plastic derivative, it can use it as a carbon source and it grows up, as you can see. Similarly, as I said, right, I'm street smart in my research and I don't have much money. So this is one thing you guys should learn. When you have your own research lab, you have to struggle for every penny to keep your research project going. So I don't have much funding right now. All I have is right now internal funding. So, but I love this project. So how can I move it? So what I did is I used my Toyota. I have a Toyota car, Corolla. So I used the 10W30 fresh and the burnt engine oil of my car as a carbon source in the TP media. And you can see it can grow when I supplement the TP media with the Toyota engine oil, actually. Then we tested some nasty coal tar derivatives, which are carcinogens like phenanthrene, naphthalene in the TP media. And then when you can see the growth of the CC4533 on these naphthalene and phenanthrene TP plates, actually. So it can use it as a carbon source because it doesn't grow only on TP, you know that. We tested monocyclic aromatic compounds like benzoate, phenyl acetate, which are common environments because of human activities. You know, we are messing up this planet Earth. We're going to destroy it someday, I'm pretty sure. So these bacteria can also use these compounds as a carbon source. Now, we tested the Clyp 185, aka Microbacterium, also on these chemicals using the TP media, but it never grew on any of these chemicals that we tested. So I'm not going to show you those negative results. But remember, Clyp 185 also cannot grow on regular tap media from, the same media from where I isolated it. So remember, I'm going to touch base on that mystery at the very end of the talk, why it cannot grow on a tap media when I isolated it from a clammy tap plate, right? So in the literature, I found that Microbacterium can tolerate heavy metals, but there is no literature available that shows anybody has tested the heavy metal stress tolerance of Sphingobia species. So we decided to test 6 heavy metals, different concentrations ranging from 0.5 to 20 millimolar to see if both Clyp 185 and CC4533 can tolerate these heavy metals. So as you can see, this is LB media supplemented with different concentrations of cadmium, and CC4533 can tolerate 0.5 millimolar of cadmium, which is pretty much 18,000-fold higher level of cadmium than the maximum cadmium level allowed in drinking water. Clyp 185 can tolerate up to 0.5 millimolar, and I mean, it grows somewhat on 1 millimolar. So it's pretty much can tolerate 37,000-fold higher level of cadmium than what is allowed in drinking water. We tested cobalt tolerance too, and as you can see, CC4533 can tolerate 2,000-fold higher level of cobalt than the cobalt level allowed in drinking water. Clyp 185 can tolerate 0.5 millimolar cobalt. Okay, my pointer went away. Okay, I can see it now, sorry. So I'm going to go quickly. For zinc, you can see CC4533 can grow. It's growing slowly, but by 28 days, it can tolerate up to 15 millimolar zinc. That's a lot, actually. Clyp 185 can only tolerate up to 1 millimolar zinc. For copper, CC4533 can tolerate 2 millimolar copper, which is about 300, almost 400-fold higher than what's allowed in drinking water. Clyp 185 can only tolerate 0.5 millimolar of copper. We tested also nickel tolerance, and you can see CC4533 can tolerate easily 12 millimolar of nickel, and it also tolerates but grows very slowly in the presence of 20 millimolar nickel, which is like 44,000-fold higher than what is allowed in drinking water. Clyp 185 can tolerate about 2 millimolar nickel, okay? Then lastly, we tested hexavalent chromium. It's a potent carcinogen, hexavalent. The trivalent chromium is non-toxic. So CC4533 can only tolerate 2 millimolar hexavalent chromium, whereas Clyp 185 can easily tolerate 4 millimolar hexavalent chromium, and it also grows slowly, but definitely survives when it's grown in the presence of 6 millimolar hexavalent chromium. So these are the, pretty much the published results that I showed you. So the take-home message for the published result is we isolated 2 novel bacterial strains, one of Sphingobia strain and the other is Microbacterium strain. And we also identified the antibiotics that will inhibit the bacterial growth, but not the clammy growth on the tap media plate. We found that both these strains produce important carotenoids. CC4533 can use toxic organic compounds as carbon sources. And unlike CLIP185, CC4533 can tolerate really high levels of zinc, nickel, copper, and cobalt. CLIP185 can tolerate higher levels of chromium and cadmium compared to CC4533. Now, we have recently sequenced the entire genome of CC4533 and CLIP185. We are actually analyzing the sequencing data before we submit it to the NCBI, to the genome bank, so that scientists across the globe can access it for their research. But we first have to know what is out there in the genome, what kind of enzymes the genes are encoding for heavy metal tolerance or for, carotenoid production or, antibiotic resistance. So this is in the very much early stage. So this is one of the major ongoing experiments. Of course, we have not published these things that I'm going to show you right now. It's a developing project. And the second most important goal that we are actually focusing all our energy and resources is to identify a nutrient media composition and abiotic stress condition that will allow optimal growth as well as minimum or maximum carotenoid accumulation. Now, you might be surprised. Why do I want to know which condition will allow minimum carotenoid accumulation? Because carotenoids have industrial importance, so I should be only focusing on maximum accumulation of carotenoids. There is a reason, Basic research is very important and it can give you some clues. So why we want to find a growth condition or environment that is under which the cells accumulate maximum or minimum carotenoid is because of two specific reasons. Number one is, everybody knows carotenoids are antioxidants and they protect the cells from stressful conditions. But are all stresses equal? And especially Sphingobia and Microbacterium, these are non-photosynthetic bacteria. They cannot do photosynthesis. So not much information is available about specific stress combative functional role of carotenoids in non-photosynthetic bacteria. By that I mean, let's say you subject the bacteria to different stress, like highlight stress or salt stress. Is the carotenoid in the bacterial cells important for cell survival under all kinds of stresses in a general fashion, or is it stress specific? So how do I design my experiments? If I find a nutrient condition, for example, I have drawn it in a picture on the slide, you see? Let's say I found a growth medium X, which supports growth and pigment accumulation. And then I find a growth medium X, which supplemented with glucose, it grows, but doesn't produce the pigment. How can I exploit this growth phenotype without resorting to genetic engineering? Because I don't want to use genetic engineering. That's the norm. Everybody is doing cloning genes, overexpressing genes. I want to use, what is naturally present in the bacteria. So pretty much I have found two conditions. One condition, it makes carotenoid. The other condition, it grows, but it doesn't make carotenoid. Now I can exploit these two growth media to test different abiotic stress to see if carotenoids are needed for survival under these stress. So the bottom panel shows you the experimental design. So let's say I plate the cells on this growth medium X and growth medium X plus glucose, the bacterial cell, and subject it to highlight stress. And after the incubation period, I would expect some colonies to show up. If I see there are more colonies on the growth medium X and hardly any colonies on growth medium X plus glucose, then I know that the carotenoid is helping in the survival of the cells under highlight stress. But let's say I take another stress, high salt stress. So I again plate the cells on these two media and of course subject it to salt stress by adding different concentrations of salt. And after the incubation period, if I find there are pretty much similar number of bacterial colonies on both plates, I know that the carotenoid is not necessary for survival of the cells under high salt stress. So in this way, I can decipher the specific stress combative functional role of carotenoids without resorting to genetic engineering. And the second objective, as you all know, must have guessed, if I find a growth condition and a stressful condition that maximizes production of carotenoids, it might have applications in the industrial production of carotenoids. So we are testing different organic and inorganic nitrogen source right now to see, what is the effect of these nitrogen source on carotenoid accumulation and growth. In the first round of experiment, we tested an organic nitrogen media, which is bactopeptone, and the minimal nitrogen media, which is inorganic nitrogen, pretty much the M9 media. And we use these two media with and without yeast extract. If you can, sometimes my pointer is disappearing, I cannot see. So YE is yeast extract, and yeast extract supplies usually vitamins and some amino acids and trace amount of carbohydrates in it. So we tested the growth of CLIP185 and CC4533 without and with yeast extract in the presence and in the absence of glucose as a carbon source. So let's compare the results of the two strains. So when CLIP185 and 4533, we can see they grow nicely on the bactopeptone and they accumulate pigment in the absence of glucose, which is the top panel. It doesn't matter whether yeast extract is there or yeast extract is absent. But the bottom panel has glucose in it. Whenever you put glucose, you can see CLIP185 doesn't accumulate any pigment, and its growth rate also is low, but it grows anyway. For CC4533, the growth rate is fine. It doesn't change, but it is pigment deficient, as you can see, slightly. But we saw this drastic phenotype change for CLIP185 on bactopeptone. We did the same experiment on the M9 media, which has minimal nitrogen source, and modified M9 media, which means M9 is the minimal media, right? If I add yeast extract to it, which is supplying some amino acids, vitamins, and trace amount of sugar, it becomes modified M9. So again, if you look at the growth phenotype, CLIP185 on M9 media without or with yeast extract, and the CC4533 growth without yeast extract and with yeast extract. So without yeast extract, of course, you don't expect the strains to grow because there's no carbon source there, because the top panel doesn't have glucose, right? And when you add yeast extract, this bacteria can grow, although there is no glucose available right now. The bottom panel shows the result in the presence of the glucose. So G is glucose in this legend. So when you add glucose, you see the CLIP185 is again white in color in the absence of yeast extract. In the presence of yeast extract, it is growing nicely and it is accumulating some pigment, trace of yellow is there. But CC4533 is completely bleached pretty much when you add glucose, whether you have yeast extract or you don't have yeast extract. And the growth, it's growing fine. So you can see the difference in the growth phenotype of these two strains in an organic nitrogen media and a minimal media like M9. Then we tested 3 other organic nitrogen media. One is the LB media with extra tryptone, 1.5% tryptone. The other is proteose peptone. And the third one is tryptic soy agar, which has milk protein casein digest in it, as well as it has the soybean extract in it. So partly plant and partly animal organic nitrogen source. And if you look, so on the CC4533 growth result, you can see it's happy on the LB with tryptone, TSA, whether glucose is present or glucose is absent. Because the top panel is without the glucose. The bottom panel has glucose, okay? So in all the, I mean, it's accumulating pigment as well as it's growing nicely on TSA and LB. But on the protease peptone, it is growing nicely, but it is bleached. I mean, it's not making pigment. I mean, it's very little, if at all, when you add the glucose to it. Or when you don't have glucose, it's also not making that much pigment. So when we looked at CLIP-185 on the same type of organic nitrogen media, we saw the best media for the CLIP-185 growth is the TSA media. Whether glucose is present or glucose is absent, it makes no difference. But when you spot it on the LB tryptone and LB tryptone plus glucose, you see a difference. Again, it's not growing very well in the presence of glucose. That's very surprising. In all these results, you see CLIP-185, when you add, grow it on LB tryptone or bactopeptone in the presence of glucose, it doesn't want to grow and it's like whitish in appearance. And the protease peptone, it grows, but again, there's reduction in growth and pigment accumulation when you add glucose. So there's something about glucose and the nitrogen source going on that is having an effect on the pigment accumulation and growth for these two strains. So we tested very recently two different other sugars, not glucose this time. Look, this is lactose and this is sucrose. And we added it to the bactopeptone media to see if we are going to see the same kind of result that we got with glucose. And we saw when we use lactose, both strains are fine on bactopeptone. It's growing, there is pigment accumulation. But when you use sucrose, the same result that we got with glucose, we are getting it. Again, it's not growing very well. And the CLIP-185 and CC4533 is growing, but it's a little bit bleached. So the only explanation that comes to my mind is if you studied genetics and also cell molecular biology, you know, the glucose transporter, the phosphotransferase system that is responsible for uptake of glucose also takes up sucrose. The same transporter is helping to take up sucrose and glucose. Whereas the lactose uptake in bacteria is mediated by a different transporter. So the results have actually highlight that, you know, the use of different type of transporter for different type of sugar uptake. Now, remember I told you that I'm going to address the point why CLIP-185 cannot grow on tap, although I isolated it from the tap media, right? So my undergraduate student recently did an experiment. She took the cells of the CLIP-185 from an LB plate where it grows nicely and streaked it on a tap plate and on 3 different tap plates, which are supplemented with tryptone, different amounts of tryptone, as you can see. And guess what? As soon as you add like 0.25% or 0.5% of tryptone, it starts growing up. And then she did another thing. She took this kind of white growth. There's a little bit of white growth. I hope you can see that on my slide. So she took those white cells and put it back on a fresh tap plate, and it never grew back. It's dead pretty much. So the result is telling us that CLIP-185 needs extra nitrogen to grow on tap. So we think we have to test it. When it was isolated from a contaminated Clammy tap plate, right? Maybe the Clammy dead cells or Clammy was giving some extra nitrogen to CLIP-185. And that's why it could grow with Clammy, but not when it is streaked by itself on a, allowed to grow by itself on a tap plate where there is not much nitrogen, unless you supplement extra nitrogen. So this is also very interesting to us. And another thing, again, this is covering the slide. So look at the color, it's yellow. This has acetic acid as a carbon source and tryptone as a nitrogen source. So what did I show you earlier? Let's go back to the tryptone result. See, this media, it had tryptone and glucose in it, right? So what happens is when you have tryptone and glucose, it cannot grow. And and it's kind of bleached. But when you add acetic acid and tryptone, it's growing profusely, and of course it's yellow in color. So we need to do more experiment on this, but this is just an observation. So I'm seeing that my audio is giving me a message that audio is weak or something. So we will end now. I just want to tell you a plan that I have for future, and that plan is based on how different biochemical pathways interact with each other. Okay, because biochemistry is very important for my research objectives. So I'm not going to go and talk about the details of the pathway. I'm just going to broadly talk about it. Now, in non-photosynthetic bacteria like Syngobium and Microbacterium, the carotenoids are synthesized via the MEP pathway. Okay, and the plants also have this pathway to make carotenoids. And the substrate for this pathway is pyruvate and GAP, which comes from glycolysis, right? Cellular respiration. Now, glycolysis also provides 3 and 6 carbon sugars that are shuttled into the pentose phosphate pathway. And recently there is a paper that has shown that xylulose 5-phosphate, which is an intermediate in the pentose phosphate pathway, actually stimulates the isoprenoid biosynthetic pathway, the MEP pathway, and that leads to the production of carotenoids, actually. So if you mess up glycolysis, or let's say you mess up glucose uptake or glucose consumption, then glycolysis will be affected, pentose phosphate pathway will be affected, and carotenoid biosynthesis will be affected. Now, for growth, you need carbon and nitrogen. These are key things, right? Because we want good growth and either good carotenoid accumulation or poor carotenoid accumulation, right? So we also have crosstalks between amino acid biosynthesis and other metabolic pathways like, Krebs cycle, glycolysis, pentose phosphate pathway, and so on. So you can see these cellular respiration phases, as well as pentose phosphate pathway, provides carbon skeletons for many amino acids that you see in this picture, which I took from a biochemistry textbook. So this tells you if you actually mess up, if you have, like, there's a problem in carbohydrate metabolism, you can also affect the amino acid biosynthesis, and that would lead to poor growth also. So there's a complex, crosstalk going on between nitrogen metabolism and carbon metabolism, and there are regulatory mechanisms that actually control catabolism of amino acids and carbohydrates. So here is another picture. So we think, these days many people say, right, high protein diet is very good for you. So eat more protein and less carbohydrate, low carb. But if you look at biochemistry, nothing in excess is good for you. So you see how these 20 amino acids, they can enter, when you eat too much protein, these amino acids will enter into cellular respiration. It's not like glucose only we eat to live, right? So fats, amino acids, lipids, they all enter into different stages of cellular respiration. Their entry point is different, okay? So here, this picture is showing you how different amino acids can enter into cellular respiration, like at the point of pyruvate, all these alanine, cysteine, glycine, serine, threonine can enter. Alpha ketoglutarate is another entry point, succinyl-CoA, oxaloacetate, and so on. And then there are other amino acids which are highlighted in yellow, which are ketogenic amino acids. They enter at the point of acetyl-CoA or the ketone bodies. So there is a paper in 2019 that has come out and has shown for E. coli, which doesn't normally make carotenoids, but can be genetically engineered to produce carotenoid. If it is grown in the presence of these amino acids that generate pyruvate or oxaloacetate or alpha ketoglutarate, what they have seen is the E. coli cells, they will use these amino acids for energy production, although glucose is present. So you're growing the cells in the presence of these amino acids plus glucose, but the cells somehow fail to take up the glucose and glucose consumption is reduced. Still, all the amino acids are converted into these cellular respiration intermediates and used up for energy production. So this was very interesting observation to me. And you saw how the results of the growth phenotype and the carotenoid accumulation with different nitrogen sources and carbon sources that I showed you. So the next thing we would like to test is the effect of these glucogenic and ketogenic amino acids and also these organic acids as a carbon source to see what kind of effect they have on carotenoid accumulation and growth. Now, okay, these are all kinds of experiments, but what ultimately I'm going to go for is the results of all these different amino acids, nitrogen source, organic acids, or carbon sources will give me some idea about what type of nitrogen source and what type of carbon source are preferred by these two strains for carotenoid production. And also the stress, abiotic stress experiments will give me the stress condition that I'm looking for. So it will help me ultimately for a sustainable way of finding an agro-industrial waste that will be rich in those organic, the nitrogen source and those carbohydrate have similar composition that I'm finding in my lab that will not only be used as a renewable energy source for the bacteria, but also used for commercial carotenoid production. So I would, my lab mates or team members, they are all undergraduates in this project. I didn't have a master student in this project. So this is Miss Taylor Berry. She's the high school kid who joined my lab. And because of this kid, I have this bacterial project. And by the way, she also isolated another third bacteria. It would have been too long presentation if I included 3 bacteria. So I selected only 2 to present. She's a co-author in 3 research publications from my lab. And here are Seth, Taylor Box, Jesse, Kevin, and my current research student who is testing all the nitrogen and carbon sources, Ellie Puckett. So without their motivation and their hard work, they work over the weekends and the nights, and they are a full-time student too. So I wouldn't have achieved whatever little I have achieved so far. And the project was funded internally by West Georgia. And of course, I must thank Clammy, baby, right? Because so far in my life, in my 25 years in the United States, I have abused Clammy for my selfish research, abusing it to create mutants and doing research on photosynthesis. But Clammy, in the end, gave me a wonderful microbiology project, which will last till my retirement date, actually. And other scientific, peers, I would like to thank Erin for giving me all the biochemical reagents for microbiology tests, Dr. Fujita, Dr. Khan for giving me heavy metals and the organic compounds that I tested on CC4533, Dr. Gary Rufkin for giving me the Pocuria bacteria strain, and Dr. Wendisch from Germany giving me the Corynebacterium patented strain, which would have cost me $300 if I wanted to buy it. He was very nice and kind enough to ship it to me, although it was patented. So I have the floor for question, and it was a long talk, and I woke you up all early in the morning. So I'm going to stop sharing my screen. And if you want to contact me, know more about the biology program, the master's program, please feel free to go to my lab website or contact me directly by email. And I would be happy to help you, or answer any questions you might have. So I hope I didn't put you to sleep.
No, madam. It was really interesting.
No, madam, no.
Okay.
I
No,
was
madam.
afraid,
Student.
although I selected 2, I had 3 bacteria. The other one doesn't make carotenoid, but it's very interesting. It makes pyomelanin, melanin skin pigment. So it makes pyomelanin, which is used in industry for metal chelators. And it also can be used for bioremediation. It can tolerate heavy metals as well as organic, toxic compounds. But that is a different project. It's with carotenoids. So please, students, I'm so impressed that so many of you have joined this webinar at such early hour. And even if I bore you to death, you wouldn't admit that because you're shy, I'm pretty sure. But, I want you to, now ask me questions, or the question can be linked to your future career. And we're here to, see if I can help you in any way. Also, you must have understood what research is, right? You start with something and the story ends up in a different direction. Research. Somebody has done a search before you. You are researching it. And the story can develop unexpectedly, like a high school student project has shaped up like this. So I wouldn't have believed it myself if anybody told me. So please, I have talked enough. I'm going to drink some water now and shoot questions.
Thank you, madam.
Hello.
Hello.
Yeah, can you hear me?
Thank you very much for your elaborate talk and just insight into a new dimension, how concerted effort, not by just a graduate student, but a high school student also. The curiosity itself is science, and it generates a scientific temper. And all our students and faculties, young faculties should understand that it is not that what you have been trained during your PhD, you should pursue throughout your life. You should have a carving to go inside a real problem. Real-time problem is more important, and that gives more interest and dividend at the end. And here, your story of that high school student and the development of a new dimension into your research field itself is a really wonderful thing. I always give an example of the Nobel Prize of double helix. It was not by any outstanding a very PhD or postdoc students, but just graduate students who have got Nobel Prize winning work. Like that, high school students in the US or Europe can do it. And this is what I have been telling my students and my faculty. Why we cannot train our high school students onwards or +2 students to work. And your story also gives me another insight into young people's mind that nothing is limiting. You don't have a grant and you say that I don't have a grant, how to do research? Yeah. Research is not done by grant only.
You have to be street smart. That's what I say, street smart, not book smart. That is very much needed because students usually get very bookish and they only think about grades, like I have to get an A. But are you really learning? Getting an A and learning doesn't go hand in hand, really.
That is what I always say, a laboratory which switches off its lights after a certain time, it is not a laboratory, it is just an office. And laboratory where day and night work is going on is really enjoyable. And that is a laboratory where something new and novel can come out. And this is the spirit of science, which you have really made this day wonderful by giving an insight thought into how to go deeply into a scientific research and develop a innovative research, not simply a dogmatic or a straight line research. Research can just branch out from one small thing to a umbrella which can grow out of proportions what we have started and where we end. So it is not that you started with something and you are after 20 years you think that I am not concentrating, but that concentration that you have made initially has given you new dimensions and new thoughts, and that new dimension and new thought is real research. Thank you, madam, for your
I want the students to ask me something. I would love to see if they have any questions, or if they have any questions about their career. Science is interdisciplinary in this today's time. There is no such subject like biology and chemistry and physics. there is no standalone science subject. It's all interdisciplinary and integrated science. And observations are very important. You see, with small observations actually helped me to design the next set of experiment. So for science, observation is critical. And if you have a passion and fire in your belly, you will make it happen somehow. You will reach out to people like I reached out for those 2 strains from that Harvard professor, and they were very nice. People are usually helpful, but you have to be proactive and you have to reach out to people. You cannot be shy. If you want this thing to happen, you make it happen. That's what I would say to all the students. So if you want to come to the United States for higher studies, reach out to people. Okay, look up you are techno savvy, right? You Google, you Bing, and you look up professors. What are they doing? Contact them, and you will get a response. So don't feel shy. Okay, don't think, oh, I'm in some small university in India, so I should sit quietly. Nobody will read my CV. Just a chance, contact people. So any student who has a question.
Professor Mitra, I have one question. Actually, some comments I have. It's really nice the way you have presented from a small problem, how you took it to the next level. I really admire that. I'm not a student, but I'm always learning. So always a student.
Me too. I am learning. I need help for the bioremediation. I have no background in bioremediation, but
but the way you presented, actually few things always hit me, and I was little bit moved, and the way you were citing different chemicals, different things, being a chemical engineer and with some background in chemistry, something actually hit me. Like these microbes, they are producing carotenoid, right?
Yes.
Now, carotenoid is something that helps the bacteria survive from oxidative stress.
Right, absolutely right.
Now, oxidative stress and also from light. So if there is a light stress, that will also help.
Yeah, so here is the thing. I didn't want to, because I was already giving a so long talk. So oxidative stress is the key word. Now, different abiotic stresses like highlight stress, salt stress, reactive oxygen species like hydrogen peroxide, superoxide anion will generate oxidative stress. So abiotic stresses generate oxidative stress, which are reactive oxygen species. So yeah, go ahead.
Here, what you already mentioned. So basically, if there is presence of free radical in the system, or you have species containing double bonds, there is always a chance to produce these carotenoids more. So basically, what I was thinking of using hydrogen peroxide, hydrogen peroxide will increase the amount of carotenoid in the system. And you were mentioning about lot of amino acids that actually help. So the common thing there is C double bond OH. So there is again a double bond O. And might be there is some tautomerism or whatever.
Right. And it is very complicated. I didn't want to show you the different types of carotenoid because I have the genome sequence, right? So I've seen the genes already, what kind of carotenoids they're capable of making. So they make some unusual carotenoids also. The genes are there, like the hydroxylated version other than zeaxanthin or the keto hydroxylated carotenoids. So there's some interesting carotenoid genes present in this bacteria, which I didn't have time too much to, because I'm also genome sequencing, right? I sequence the whole genome. So yeah.
And also these carotenoids, because I don't saw all these things, but what strikes me that carotenoids show color. That means it should contain conjugated double bond.
Right.
Now, if the stress is more, then conjugation should be more. So if the stress is, then the free electrons, the energy required for that will go down. So more conjugated double bond will help in just surviving in lot of stress. So that's why what I think of using these amino acids, which has double bond OH, and also these keto compounds, C double bond O, all these things will really help in this. And another application, what I thought, you mentioned about anthracene or phenanthrene kind of stuffs you mentioned.
Yes, coal tar derivatives. Yeah.
Yeah, actually, I used to work in Canada on this aluminary, so for carbon anodes, and there is a huge problem with the starch.
Okay.
When you bake the anodes, there is this naphthalene, phenanthrene, anthracene, all these things are coming out and which are pretty much carcinogenic. So
Right.
what I was thinking as an application of this bioremediation part, because I noticed it works very good against these species. So if we can channelize these stars into these kind of bioreactors, then it will be of immense use in aluminium industry, because this pollution during baking is a major challenge there.
Mm-hmm.
In Canada, actually, I used to work with aluminary alouette, and maybe in US you can connect Alcoa.
Mm.
So they are really struggling with this.
You know what? Although my title said, they have potential, it's all in a lab setting what I have done so far. So, there are different ways to do this in situ as well as ex situ bioremediation. I'm looking for, collaborators actually who can do it, because I cannot do it. I don't have that kind of background, but I'm willing to, share the strain. And obviously I deposited Clip 185 strain to the culture center, but I have not deposited the CC4533 yet and the third bacteria. I'm looking for potential collaborators who will be able to test out these bacteria,
Maybe
the field site or something.
we will be happy to collaborate actually, because I have some experience in those areas. I have worked a bit, but I will be happy to collaborate. But the way you presented, it's really praiseworthy. I must say that
I'm in a very small university with meager resources, but I have some very motivated students and who are not scared of hard work. And so I'm blessed in that aspect. I always get the best students in my lab, actually. And I have fun with that, with them. So I thank God for that. But yeah, I'm very happy how the story has shaped up, and it's going to go, because I have a lot to analyze the whole genome sequencing data, And I actually haven't submitted it to NCBI because I want to see myself what is there before I submit, actually. And I do need help for that also, because I've never submitted a genome sequencing data. Just a partial gene, yeah, it's easy to submit. But the entire genome sequencing data, I've never done it. So I'm also looking for a person to sit beside me to help me through the steps.
So
I think
that is
NCBI does
possible,
offer
right?
help. Yeah,
Like
there's a
here
lot more things to do, and I need graduate students. But here, usually the graduate students, it's very hard to find a good, motivated, hardworking graduate student. So yeah, if anybody's interested, I would say my university is small. It's close to Atlanta, very cheap cost of living in Georgia. So if you are daunted by United States, you should come to a small place first, get adapted, and then move to bigger schools. So this is a perfect ecological niche for students, international students who want a low cost of living, good weather, doesn't snow much at all, once a year maybe. Beautiful nature is here. And then you want city life, you go to Emory, Georgia Tech, downtown Atlanta. So it's 45 miles from my Carrollton city, actually. So I am actually looking for graduate students, thesis track, not non-thesis, who can continue. There are 3 bacterial strains. So any one of those projects they can work on. And particularly now, I have this genome sequencing sequencing data, that itself could be an in silico project, bioinformatics.
Yes.
So not even wet lab experiment, that's even less work, only computer work. So yeah, if you happen to know, if there are any students who have, please, there are a lot of chat things. I haven't checked the chat box. Oh. I haven't checked the chat. Is there any question there? Oh, the Toyota oil. Okay. Somebody has asked me about the, let me go up, scroll up. Let's see. First question, Deepak from MSc Botany. Germs are everywhere. We try to clean our homes, especially but do we really get rid of all the germs? Especially, what will be the different ways to get rid of such types of bacteria or viruses during this COVID scenario? I have no perfect answer for you. You can try Clorox. There are all kinds of, antiseptics available now since COVID started. So not all of them are 100% perfect. They say 99.99% efficient, the Clorox. So it's hard to say. And different types of microbes have different, ways to resist these antiseptics. And if you use too much of these chemicals, they can become resistant too. It's a constant chemical warfare going on between the bacteria and the humans. Same thing for antibiotic resistance. Okay, so good morning, ma'am. How the Toyota engine oil is used in medium. Okay, so how do we use those toxic chemicals? We made the TP medium, and then it's very hydrophobic, right? The engine oil. So we coated it. You saw the person, we diluted it. You must have seen those numbers on the slide. And then we took 2 ml or 4 ml, and it was dissolved in chloroform, by the way, and then coated on it, and the chloroform, of course, evaporates and it dries up. So you must have seen for some aromatic compounds like phenanthrene, there were crystals because the chloroform evaporated and the crystals of phenanthrene were left. So we need a better way to have a smooth lawn, but that's the best we could do. And it worked actually. You can see the cells growing on top of the crystal. So that's how we plated all the organic compounds. In upcoming future, can we face biowar by using? Yeah, that's what I answered already. This question from Deepak. Thank you. You guys are thanking me so much. Thank you for being here. And I was thinking you guys will be snoring by now because it's a too long talk and I don't know if the sound.
Excuse me, madam. Hello.
Hello.
Am I audible? Madam, so
yes.
As I am working on the microalgae and cyanobacteria, so while giving bacterial stress also now, cyanobacteria, that algae turns into yellow and pigmentation starts. So can we have any type of development of consortia or microalgae, beneficial microalgae and along with the cyanobacterial algae so that it will give more carotenoid contents? Because carotenoid contents are already put photosynthetic pigments, plants, they already have their photosynthetic pigments. And during stress condition, it induces the carotenoid biosynthesis pathway. So can we have any possibilities to make a consortia of beneficial microbes along with some suitable cyanobacteria or algal strain that will increase the productivity of carotenoid biosynthesis?
Yeah, but they are individually going to make the carotenoids, right?
Yeah, yes, but during stress condition, it itself also growing.
I don't know about the yields because this is a new subject for me because I've never done like commercial level carotenoid production. I'm learning too. So logically, if I have to look at the scenario, then you have two different, one is a bacteria and the other is a green algae. They can have symbiotic relationships.
Yeah.
Like I think my microbacterium was growing on that contaminated clammy plate because it was getting some nitrogen from the clammy. But when you don't have clammy, it doesn't want to grow on that plate. Then you have to add extra tryptone to make it grow. So
even if you use nitrogen cyanobacteria, it also that give a carbon source and nitrogen source to that organisms. So it will have minimized nutrient requirement is also minimized, and that case along with that bacteria give the stress for the cyanobacteria. The cyanobacteria carotenoid synthesis will more than that of the chlorophyll biosynthesis. as an antioxidant content. So it make a probability make a good consortia. So
Yeah,
for commercial production.
Actually, I saw that paper. Remember, in the very end, I showed you that there is a paper that showed that the pentose phosphate pathway, intermediates are stimulating carotenoid biosynthesis. That was done in cyanobacteria, actually.
Yes, sir.
So they actually overexpress 2 genes of the pentose phosphate pathway to get more xylulose 5-phosphate, which will stimulate the MEP pathway for isoprenoid biosynthesis, actually. So there are genetic engineering being done in cyanobacteria because I think that's the main organism people are working on other than bacteria. But to me, it is interesting. I'm a plant biologist, right? So photosynthesis person. So these bacteria are not photosynthetic, but they make these carotenoids and they use it for a different purpose to combat stress, different types of stress. Not I have to test it, the highlight stress, because if you look at the literature, these Fingobium and Microbacterium, they also infect plants, by the way.
Yes,
They're plant pathogens. Yeah. And they do not grow under very high light. They grow attack the roots of the plant. So where there is underneath the soil, so there is dim light there, very little light. And I didn't show one thing. Clip 185, the production of, and Corynebacterium, the production of carotenoid is strictly controlled by light. But the Cocuria strain that I showed, making decaptonazanthin, it makes carotenoid constitutively. It doesn't care whether light or darkness. I didn't show you that result. So there is a light factor that, of course, light stimulates carotenoid production, we all know, from photosynthetic organisms. But in bacteria, sometimes light is needed for carotenoid biosynthesis, but other bacteria, light is not needed. So they have a different way of regulating carotenoid biosynthesis than typical higher plants or algae. Any other questions? Okay. Trash bin and Toyota car. Yes, guess what? If I would have thrown those plates in the trash can, I didn't. I gave it to that. So I thank that high school student. If she didn't walk into my lab, I probably would have thrown those plates in the trash can. So she was God sent, actually. Right time. Timing is very important, right? In life. So no questions, no future career questions? Okay. So I guess I took a lot of your time. And if you need any help, please, students, contact me. You can find me by Googling or get my email address from Dr. Bhattacharya. Okay. So have a wonderful National Science Day. And I hope you have a poster presentations, right, lined up. Yeah. It's in the afternoon.
Madam,
are there any awards for the poster? Is there a competition or is it just a display of poster?
There is display and competition is also there.
Okay. Do you have oral presentations?
No, madam. Actually, exam is going on, so oral presentation is not there. So.
Okay.
Madam,
okay.
Is there any scope in your lab for our students?
I mean, as I said, master's program, if any undergraduate wants to come here for master's thesis degree, yeah, I would like to have a motivated, graduate student who is hardworking, actually. And, my undergraduate, I'm getting old also. So these days, I used to have like 5 or 6 undergrads at the same time, but now I have only one. She's going to graduate, but I'm looking for a good undergraduate student. My research is a teamwork. It's a division of labor. So, students have classes. Sometimes students work also outside school to, here the students have a very hard life, let me tell you. Like, in India, the students, they look at the culture, the American culture, like the New York and San Francisco, and they think, oh, life is so smooth, so easy here. Here, the undergraduates pay their tuition by washing dishes in the restaurant. Nobody looks at that, the hard work they do. They come with red eyes to the class next day because, education is very expensive. So they They pay for their own tuition. They don't depend on their parents sometimes. And they come from a very poor family also. There are many students. So nobody sees that aspect of the American academics, I think. So I really respect my undergraduate students. I never had to do that in my life when I was a bachelor's of science student and went to Presidency College. My parents paid. At that time, the fee was also very low. I mean, it's ridiculous fee I had in my time. But I actually learned a lot from my students, It's not like I teach my students, it's a two-way traffic. I learn from my students too. And I think all faculties should do that. They should also think like students sometimes to get into their heads. It's a different world. Once you come here, you will understand. I had a very interesting journey, as I showed you. It had its ups and downs. I had to learn it the hard way because I never had a proper guide to guide me. So that's why I understand the importance of a mentor, faculty mentor. I mean, you can be a great professor, but I'm talking about mentoring your students. So my students are really successful. I'm very proud of all my undergraduate students that I have. They're in real, they're doctors, they're professors, they're in NIH. So, and I use them as current mentors to my motivated students now. So they have kept in touch with me. So I want you students to feel free and contact me if you think I can help you. I will be very happy to help you. Even if I cannot take you in my lab, I can point you to the right direction. Okay?
One more question for students. I want to ask, you can see the chat.
Let me see. Is it in the chat box?
Yes, madam.
Okay, is it the last question? Thanks, ma'am, for inspiring us as a bacterial.
Oh, madam. Yes, madam.
Oh, radioactive. No, I cannot test that. I don't have, my university doesn't have permit to handle radioactive elements. So I will never be able to do it here. If somebody else wants to do somewhere else, and no, that is not possible beyond the scope about the radioactive elements. I don't think they will be able to tolerate radioactive elements. Yeah, we have a lot of other nasty chemicals to test. You can have a never-ending list. But I'm shifting my research in a different direction now, as you saw from the future experiment plan. So we'll see where it takes me. But no radioactive elements. Any other question? No? Okay then, thank you again all for attending the talk and for giving me this opportunity to share my research, my lab's research with you all. And maybe I'll hear from some of you via email. The students pretty much wanted to type, they didn't want to talk to me. Okay then, good night from my side and good afternoon pretty much on your side. It's almost 11:49 p.m. and I must go to bed because for a whole day teaching I have.
Good night.
Okay, bye.
Bye. Thank you very much.
Bye-bye. Good night.
I think. Santanu, is there a vote of thanks or something?
Thank you, Professor Mitra, for chairing the session. And it was a nice talk delivered by our honorable guest, or Professor Matusi Mitra. And it's time to acknowledge the efforts we all have done for making the event a successful one. So first of all, I would like to thank our own VC madam, Professor Supriya Patnaik ma'am, to wake up very early and talking about the university and making this event a success one. Like to acknowledge the effort of our own regional director, sir, Pradeep Sarangi, sir, who in a small span of time, who could able to manage the things and tried of everything and make it possible. I would like to thank our Pro-VC sir, Professor Dipankar Bhattacharya, to join the event with all enthusiasm and also asking the questions to trigger the budding minds. My sincere thanks from the organizing committee to Professor Amarendra Mishra sir, the distinguished professor of Centurion University of Technology and Management. Sincere thanks goes towards Dean, School of Applied Sciences, Dr. Yashasvi Naik, for guiding us in the right path. I would like to thank the academic coordinator, SOS Balangir, Dr. Professor J.K. Meher sir, for helping us in all possible ways to make it possible. Sincere gratitude goes towards Principal SOS, School of Applied Sciences, Sri Somnath Sarangi sir, who contributed the all possible way to make the event a successful one. My extension of gratitude goes towards HOD and faculties of the entire Cutm family who woke up early, joined the event, and make it a successful one. I'd like to thank the HODs of Odisha campus, Dr. Botany Mishra, the other faculty member, Dr. Sagarika Palda, Dr. Suhas Kumar Sutar sir, Mr. Sriman Pradhan sir, Kalpita, Dr. Kalpita ma'am, Mrs. Bhagyasri ma'am, Dr. Gyanaranjan Mahalik, for their contribution. Of course, from our side, we have done a lot to make it possible. I should not forget the efforts of the convener, Dr. Shantanu Bhattacharya, along with Namita ma'am, Dr. Pratibha Satpathy ma'am, and other team member to make it possible. I extend my heartfelt gratitude towards the HOD of Balangir campus, the HOD Mathematics, Dr. Bharat Kumar Maji, HOD Chemistry, Dr. R.C. Sarangi, and Dr. Srikant, and all team members, Tikna Madam, and the HOD Physics, Dr. Neela Mohanty sir, Meena Madam, Sasmita ma'am, Gyanandir sir, who joined it early and contributed their way for making the event successful. HOD Geology, Bharat sir, then IT department faculty, Sanjeev sir, Pratibha ma'am, JK sir, Asa ma'am, everybody open up the IT lab for support and make the event possible. I extend my all sincere gratitude towards all the family members of Centurion campuses, the BBS Bhubaneswar campus, Pallakhemundi campus, and Balangir campus, and all those who joined very early. I praise the effort made by the press and media team led by Mrs. Pratibha Sathpathy ma'am and Abhishek particularly to make the event a seamless one. Of course, I would also like to thank the technical support team who support us in every possible way to make the event a grand success one. Not the least, we should not forget the efforts made by the students. Yes, they are the real people who contributed the enthusiasm and asking questions. This Science Day is meant for them. And yes, these people make this event a successful one. Please forgive me if I have not mentioned anybody's name. Thank you all for joining and making the event a successful one. So it's time for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, madam. Thank you all.